In the previous video, we discussed about the antigen. If you want to watch that video first, the link is in the description. Now in this video, we'll be discussing about the antibody. The antibody is also known as immunoglobulin, written shortly as Ig. The antibody is Y-shaped glycoprotein secreted or produced by plasma B cells. The antibodies neutralize, control or stop the pathogens and also aids in immune response. Now first of all, let's discuss its structure. As we know, antibody is Y-shaped protein. It has got two heavy chains that makes it Y-shaped protein as shown in the diagram. On one chain it has got 4 domains, 1 variable domain and 3 constant domains. These constant domains are written as constant domain 1, constant domain 2, constant domain 3. In the same way, we have 2 light chains, but it has got only 2 domains, 1 variable and 1 constant domain. The light chain is highlighted in yellow color. So here in this diagram, this region is the variable region, which consists of 2 variable domains from heavy chain and 2 variable domains from light chain. But we divide antibody into 2 regions. It's FAB region or Fab region and FC region, depending upon its interaction with antigen and other proteins of immune system. The upper region in this diagram is the Fab region, fragment antigen-binding. It consists of 1 constant and 1 variable domain from each chain. Then we have FC region, that's fragment crystallizable region, with 2 constant domains. Then in the structure of antibody, like all other proteins, it has also got N-terminal and C-terminal ends. And we see the variable region has got unique site, what we call as paratope, which is the antigen binding site. Let's see the Fab region in detail first. The Fab region binds antigen. It has got 1 constant and 1 variable domain. The variable domain of Fab region contains paratope, which is the antigen binding site. This paratope is a set of CDR, that's complementary determining region. And we have 3 types of CDR, CDR1, CDR2, CDR3. Each variable domain has 3 CDR, which makes 12 CDR molecules for single antibody. 3 on each variable domain, that's 3, 3, 3, 3, that's 12 CDR molecule for single antibody. Now getting to the FC region. It interacts with cell surface receptor called the FC receptor. It also interacts with some proteins of complement system. This FC region allows antibodies to activate immune system. Furthermore, we have the isotypes of antibodies. We see the Fab region is hypervariable. It exists in numerous forms, whereas FC region only occurs in few variants, which defines the antibody class. That means the FC region, which has only heavy chain, designates the isotypes of antibodies. So we call it heavy chain isotypes. The isotypes include alpha, delta, gamma, epsilon and mu. The alpha isotype is the IgA antibody. The delta isotype is the IgD antibody. The gamma isotype is the IgG antibody. The epsilon isotype is the IgE antibody and mu isotype is the IgM antibody. So these are the 5 classes of antibodies. We also have light chain isotypes as kappa and lambda. But these isotypes do not designate any other class of antibody, because antibodies are classified on the basis of heavy chain isotypes only. The structure and function of each antibody class will be discussed in the next video. I hope you like the video. If you like it, give it a thumbs up. Do consider supporting my work on Patreon and also make sure to subscribe the channel. Thanks.